So, for my topic, I chose the life cycle of a bacteriophage. And so, in this video, there's a few things that will be discussed, and this is the stages of a bacteriophage when in the lytic cycle. So, in the lytic cycle, usually the host cell is killed immediately after viral replication, and there are 6 separate steps: adsorption, penetration, uncoating, viral synthesis, maturation, and release. And a bacteriophage is a virus that infects a bacterium. So, for step 1, or part A, is absorption. And this is when the bacteriophage attaches to a specific receptor by means of tail fibers that are on the end of the bacteriophage. So, in this picture, I've drawn a bacteriophage in pink, and it has a protective coating and a viral genome, and there's little tails drawn at the end, and this is what helps attach to the host cell. Step number 2 is penetration. This is also known as the injection of the virus into the host cell. The tip of the bacteriophage, as on the previous slide, you saw the pink tails that come out at the end of it. The tails contain an enzyme known as lysozyme, and lysozyme is responsible for dissolving the cell wall of the host. So it helps break open the host so that the viral DNA can enter. And there's also a hypodermic syringe-like tube at the end of the bacteriophage, and it allows for the sharp penetration of the host cell. And this can allow all of the viral DNA to enter. For step number 3, it is uncoating. And uncoating is the removal of a capsid or envelope that surrounds the virus. The capsid of a virus can be naked or enveloped. Envelope is kind of just a protective coating that is harder to break down than a naked virus would be because it blends in with our immune system cells. But there are 2 ways that uncoating can occur. One way is when the virus penetrates, the virion disassembles, so only the nucleic acid enters the host. So this is like on the previous slide, where the virus enters and it's already disassembled from the envelope. And there's a second way, and that is when the entire virion can enter the host, and uncoating will happen later once it's in the cell. The 4th step is viral synthesis, and this is also referred to as the latent period. And the latent period is when it's the period between infection and when the virus starts to show symptoms. This is the step where the viral DNA that is already in the cell starts to produce its own viral proteins and other components necessary for maturation and release. And some of the other proteins and components that the viral DNA starts to produce inside of the host cell is internal proteins, coat proteins, and lysozymes. And this is just so that new viral bacteriophages can be assembled and ready for release. For part E, or for the 5th step, is maturation. And this is a pretty simple step. It's where all the components that were previously made in viral synthesis are assembled, and they produce the new viruses that will be released later on so that it can continue to infect the host. The last step is release, and it's the 6th step. This is where the newly assembled viruses begin to exit the cell and go to continue to infect other host cells that are in the bacteria. This can occur in 2 ways. One way, if the virus is naked, meaning no envelope or anything. An encoded protein like lysozyme will start to dissolve the cell membrane and the cell wall of the host cell, causing death and releasing the new virions. So in this picture I put on this slide, you see where the cell wall has been broken and the virions start to release. And there is also a second way that these new viruses can exit, and that's if it's enveloped. If it's enveloped, it'll start to push out of the cell membrane and it'll form a bud and it will be enclosed with the viruses, but it pinches off so it doesn't end up killing the cell. So overall, this is the entire cycle of the bacteriophage, and there's usually 6 basic steps to it: absorption, penetration, uncoating, viral synthesis, maturation, and release. And each one of these steps helps to accomplish viral replication, and so that those other viruses can, spread and cause more infection. And this only occurs in bacteria when this bacteriophage infects the bacteria.